Hello, everyone. I think I'll get started right on time. I'm mostly going to introduce my colleague, Dr. Kara Statz. So without further ado, welcome. It's great to see all of you again after a brief hiatus in rare disease. It's great to be back in AMP. My name is Mark Keel. I am the Chief Science Officer and founder of Genomenon. My background is as an MD, PhD with a focus in molecular genetic pathology. Specifically, I spent a long time in profiling hematopoietic malignancies. But as I said, for various reasons for Genomenon, I took a detour in rare disease, so it's great to be back here. Just to calibrate your attention, what we're going to be talking about is genomic variant interpretation. So this has nothing to do with the wet lab side of things, producing tests or running tests. This has all to do with the analytic aspects of molecular diagnostics. And specifically, we're going to talk about leveraging comprehensive curated content and leveraging that to improve detection of causative variants and to facilitate reporting and the actionability of those variants for both somatic and germline variants in multiple different contexts, specifically in the context of large gene panels up to and including whole exome and whole genome. So obviously, Kara and I are employees of Genomenon. We're also shareholders, and outside of a pride and ownership, we have no scientific bias to declare. This is what we're here for. So these are the learning objectives. This is meant to be educative. We're here to share, give you a perspective on the products that we're developing at Genomenon, but also to give some context. And the context that I'm going to be setting here is to speak to the benefits and challenges that attend large gene panel sequencing as well as whole genome sequencing in a clinical cancer diagnostic setting. Outside of that background, Kara will be sharing how to leverage index data and curated data to streamline those workflows using that curated content that I alluded to earlier. I'll begin with that background, speaking to those benefits and challenges, and then Kara will close out with some specific examples. This is meant to be more of a practicum. She'll be speaking to use of the Cancer Knowledgebase, formerly the Clinical Knowledgebase, which many of you may be familiar with from JAX, as well as Mastermind, which is Genomenon's flagship product. And all with a focus on uncovering that valuable insight to enhance those diagnostic workflows. So I get nostalgic whenever I come back to AMP. It makes me think of my training, and I remember specifically how quaint it was in the molecular diagnostics lab when we would run single analyte assays, like restriction fragment length polymorphisms, and how far we've come as a field to be part of that groundbreaking change being brought out by genomic sequencing. So not only has the technology changed dramatically, but the applications have grown, even since they were first promised when next generation sequencing was initially brought to bear. So the benefits of genome sequencing are many. In addition to improving diagnoses, as I'll showcase in some slides, as well as improving prognosis, which was just an early development that I experienced some 15 years ago when I began my training, but increasingly can inform treatment through precision therapeutics that we're all well aware of. There's other aspects that come out of molecular profiling using genome sequencing, and that includes sequencing for monitoring disease progression, minimal disease, the development of new clones, resistance mechanisms, et cetera. And I'm happy to say, screening initiatives, so large-scale screening applications, both in cancer as well as a field that's near and dear to my heart, in newborn sequencing, to uncover disease before it manifests symptomatically. So all of those put together showcase the great benefit afforded by these applications of large-scale genome sequencing, whether it be panels or exome and genome. So a little bit of a compare and contrast. There are many roads to get to some of these actionable variants. Some are better than others, some are more practical than others, some are more high yield than others. When you reflect on those single analyte assays, we're obviously worlds away from that now in terms of our ability to scale and our ability to enhance our diagnostic yield. So specifically, there's benefit in larger and larger gene panels when you compare them to those single point assays or even smaller gene panels because you have more potential actionability. You can cast a wider net, you can streamline your internal pipelines because you don't have to have individual assays, you can consolidate that workflow under one, and then what remains is just the downstream analysis, which again is attendant to some of these challenges that we'll be talking about. So not only can you diagnose more readily, you can find more of those causative variants, but you can understand what diseases are readily applicable to those cases. Again, increasingly we're seeing variants and therapies being married to each other. And similarly, there's increasing specificity for trial eligibility. And that is often coming down to a genomic perspective. So the more genes you include on your panels, the more likely you are to open the aperture for better treating your patients. Similarly, away from single analyte assays or small gene panels, large gene panels allow you to more accurately calculate other molecular features like tumor mutational burden, or TMB. So if we extend that, the arc of the past from single to small gene panels to multiple large gene panels, all the way through to exome and genome, when I think about genome sequencing, I'm reminded of somebody after a long night at the bar who's looking for their lost car keys under the lamppost because that's where the light is. If we look by genome sequencing, we're able to find more, we're more likely to find the car keys that really drive the patient's cancer and can inform the patient's treatment. So that doesn't just come in the form of more genes, it also comes in the form of more different types of variants, including non-coding regions that are otherwise hard to capture with gene panels, including promoter, regulatory, intronic, splice region variants, et cetera. And similarly, given how you have to prepare for these large gene panels and the way that the sequencing comes together, understanding these large structural variants, fusion events, as well as copy number change is facilitated through whole genome sequencing. So there's a bit of a compare and contrast, but in the main, large gene panels up to and including whole genome sequencing is beneficial to the patients, but as we'll talk about here, it comes along with increased complexity in analytic workflows. So I mentioned earlier that my bailiwick has, up until this point, been focused on rare disease. This is an example, for those of you who may not be so familiar, where genome sequencing demonstrably increases diagnostic yield. So this is from a 100,000 genome pilot on rare disease and healthcare from a New England Journal of Medicine paper published recently. The graph is complicated, but on the y-axis is diagnostic yield. So how many patients get a meaningful diagnosis that's informed by genome sequencing? The bars obviously reflect that diagnostic yield for individual diseases. So there's a lot of heterogeneity across different types of diseases, but when you look at that as a whole, about a third of patients with rare disease benefit from genome sequencing when they have a monogenic cause, and an appreciable fraction of patients with a very complicated case benefit from genome sequencing. And critically, even though it's rare disease, about a quarter of those individuals have some action that can be taken. So outside of just being diagnosed, there is a treatment. That treatment may be pharmacologic, it may be nutritional, it may be interventional. One of the things I learned in my clinical training is patients and their families benefit sometimes greatly just from having an answer. But we can go further now even for a significant fraction of these rare diseases and inform patient care. We're here to talk about us being able to do that too in cancer. So the examples are legion. This is one example from a recent poster in the Annals of Oncology from ESMO. I don't have the averages here, but each row is a specific type of disease, and the columns there are showing the proportion of those cases that have actionable variants that are uncovered from a large genome sequencing platform. What's being illustrated here, if you look at those two middle columns, is a panel comprising 50 genes has a certain recovery rate or diagnostic yield, but a gene panel comprising 315 can increase that diagnostic yield. So that may be obvious, but the data is clear. There's great benefit in expanding your analytic search space to include more genes through larger and larger gene panels. And we're talking about actionability, so obviously that comes along with drug associations for those variants, whether on-label or off-label. This is another one of those examples. It's more specific. It's actually the field that I used to work on, which is hematopoietic malignancies. It's basically illustrating the case that whole genome sequencing for these individuals can inform care. A fifth of these patients had care that was changed solely because of whole genome sequencing. And again, that comes along with not just the diagnosis, but ascribing an appropriate care. So genome sequencing doesn't just uncover those variants, it informs the care. Another case to be made here is that it's increasingly relevant. This is not a stasis. Those numbers that you saw are not going to stay, they're going to increase. So there's going to be an increasing differential benefit to sequencing more and more genes. So this is a result, again, one of many from a Nature Genetics paper profiling about 10,500 cancer genomes. And the long and short of it here is the yellow and the dark blue in the stacked bar chart there are loss of function and gain of function variants that are newly uncovered through this large-scale sequencing. So this was a research assay, but each one of those variants in 300 genes, including 74 that were novel to any cancer, the balance of that are genes that have not been previously associated with a specific type of cancer. We're learning. So there's more information that can be gained from these larger platforms. And the last figure there is actually illustrating how that will benefit going beyond the 30 to 50% or so of patients who received an actionable diagnosis. We're going to see that number tick up. In this case, it was 55%. There's problems, though. The challenges here, as I mentioned before, is a more complicated workflow. So germline variants can be very beneficial. When you see a germline variant in a clinical cancer case, it may impact the prognosis, it may impact the care of that patient. It certainly also has ramifications for that patient's family. In a talk earlier, there was a figure that was cited that was much higher than 10%. It was actually 17% of cases have attendant germline causes for at least one of the variants in those cases. So understanding that, matching the sequencing to a normal to confirm that it is in fact germline, that can be impactful to care. We're going to increasingly see those as we learn more and more about how germline variants cause disease, and that further complicates the interpretation pipeline. So I mentioned that those germline variants are trickier. They warrant some high-touch attention. There's also this specter of, that undoubtedly many of you are familiar with, the variant of uncertain significance. So this is a scourge in rare disease. We see this all the time. In this case, for cancer, the number is, the proportion of variants that are VUS is about a third. In rare disease, it's much higher. These are variants of uncertain significance for which there is insufficient evidence to determine pathogenicity unequivocally. There is nevertheless frequently some suspicion that they may be disease-causing, and particularly where you can marry the variant and the disease causation to an appropriate treatment, understanding whether they are VUS or VUS in waiting, also referred to as hot VUS, as opposed to tepid or cold, which tend to be closer toward benignity. This is important, but it's challenging, and it's purely interpretive. Just to really further underscore the challenge, this is a slide illustrating the burden of existing knowledge. So I alluded to several research studies. Information is proliferating. Insights abound. Coalescing those insights, making sure that they're harnessed and that they can be figured into these workflows efficiently and accurately and with a view toward actionability, that's the challenge. So without reading through all of them, it has to do with drug approval data, variant pathogenicity, conformity to guidelines, diagnostic data, including clinical data from the patient or the patient's case, prognostic data, trial data, et cetera. All of that needs to be figured into this funnel as you're interpreting these variants to divine which of the variants are causative, which of them will inform patient care. And critically, and what we're here meant to talk about, is pre-curation of that data, preordaining what is important and being able to align that pre-curated evidence with the patient's case. That can expedite workflows. And that's what Genomenon does. As I mentioned, I'm the Chief Science Officer and founder of Genomenon. We are a 200-person company. We provide genomic intelligence. We curate, we index this data computationally, we curate it expertly with our team of human reviewers. We do so to inform clinical diagnostics as well as precision therapy development with partnerships through our relationships with pharma. But writ large, we simplify complex genetic data into actionable insights. So this is a slide that speaks to the company's history, founded 10 years ago, came out of stealth mode 3 years after that, began working with our pharma clients 2 years after that, as I alluded to, working with large-scale countrywide genome sequencing projects in rare disease. Have since that time, about 2 years ago, we acquired a large team of curators to bring to bear in the service of our mission of curating the entire genome. We do that in two ways, one at the gene level to identify those gene-disease relationships that I alluded to, but also very detailed curation of evidence and actionability at the variant level. So those two levels that required having a team of experts to be married to our AI and computational technology. And that was a long preamble to bring up why we're here, what we're here to talk about at AMP. Less than a year ago, we acquired the talented team and the valuable data asset from the JAX clinical knowledge base, now the Cancer Knowledgebase, because in our effort to curate the entire genome, we realized that we had a long row to hoe in front of us if we were to tackle somatic, and the good people at JAX had done an expert job harnessing that data, and it just made sense to align. So there's a great combinatorial value of bringing those two groups, the CKB group and the Mastermind group at Genomenon together. I want to spend a little bit of time talking about this to show you that complementarity here. So the Cancer Knowledgebase, formerly JAX Clinical Knowledgebase, is a comprehensive data set across 2,000 genes of expertly curated information for variant pathogenicity, actionability, FDA drug labels, targeted therapy, functional studies, trial information, et cetera, that Kara will go into greater detail about. It's a data offering as well as a software offering. So the focus and the content of CKB is on actionability for somatic and germline oncologic variants. You may not be familiar with Mastermind. It's another reason to have assembled you here. Mastermind is a reflection of a comprehensive indexing of the entirety of the scientific and medical literature. So every article published that has to do with genetic disease, genomic variants, and related terms, that has been indexed by our computational intelligence, looking for genes and variants and how they associate with phenotypes and diseases and drugs. It's meant to be maximally sensitive and present the evidence to our users who tend to be variant analysts. I mentioned, though, that Genomenon has a mission of curating the entire genome. Those curators, numbering about 100 that I alluded to earlier, they have been systematically curating that data, reviewing that evidence, passing judgment, and showing their work at the gene level and the variant level, including interpreting those variants with provisional calls to inform some of those pipeline automations. So Mastermind is broad. It covers all genes and multiple different inheritance patterns, including somatic. We've curated a lot of the rare disease genes that I'm talking about, the constitutional or germline diseases, but there's a complementarity between the CKB data, which is somatic and focused on action and expertly curated by the CKB team, and the index data in Mastermind for rare disease that has been curated. So last slide before I bring on Dr. Stats. The two data sets, the two resources are valuable depending on what the lab that you're running is focused on. So labs that are focused on somatic panels, or in the context of oncology, run whole genome or whole exome panels, CKB is the first place to go to, as Kara will illustrate. It's curated for functional studies, for variant pathogenicity, for drug approvals, guidelines, clinical trials, et cetera. Mastermind, it can be used as a follow-up. It has ancillary data that can help clarify some of that information, get you to the primary source of that information to allow you to go deeper, as well as to focus on uncovering some of those more rare variants, more obscure variants. In contrast, labs that are focused on hereditary cancer panels may first go to Mastermind, where, again, the focus had been on curating constitutional content, including hereditary cancer genes, some of which are part of the ACMG secondary findings that you may be familiar with. However, CKB has germline content as well and is, as I mentioned, very focused on actionability and some of that drug and trial data that Kara will allude to. So without further ado, Kara, I invite you to the stage. Thank you for your attention.
Okay, great. Hi, everybody. My name is Kara Statz. I'm one of the senior clinical analysts that works on CKB, which I've been doing for almost 10 years now. So I'm going to walk you through a few case studies highlighting how you can leverage CKB and Mastermind together to uncover valuable insights and streamline workflows, really identifying the pertinent information to a patient that could aid in clinical decision making. So looking at our first case, we have a patient with neurofibromatosis type 1, diagnosed with that, as well as metaplastic breast carcinoma. So specimens were submitted for testing, including the tumor sample, as well as a whole blood sample for matched control. So really, what is, the is there a potential germline variant underlying that neurofibromatosis type 1? And are there any variants that could be considered actionable for the patient in terms of their breast carcinoma? So samples were sequenced using a large gene panel. And here we have the results. So germline, there were 2 NF1 variants, including a synonymous missense variant as well as a non-synonymous missense variant. And then somatically, there was 1 BRCA1 variant and 1 BRCA2 variant, and both are non-synonymous missense variants. So if we start with first Mastermind, looking at those germline variants, looking specifically at the synonymous variant. So we can use Mastermind, again, to interpret germline variants with the curated evidence that you would find in Mastermind. So looking to the left here, we searched on L234L, and you can see a screenshot basically of what you would find for that particular variant. Now, if we focus on the top left-hand corner here, looking specifically at the ACMG interpretation. And so here's all the criteria that was used to support the classification of this variant. And in this case, it's actually classified as benign. Below that, you see some population data. And again, this is showing basically from over here on the right-hand side here. So it's just kind of zoomed in here. But you can see that this particular variant is very common, and this is based on NOMAD data. In addition, you see ClinVar variant calls as well within Mastermind. So in this case, this variant, you can see it right below here where it's classified as benign. You can see that it is classified as likely benign in ClinVar. And you can actually link out to the ClinVar record by clicking on this link right here. All right, so our first variant, this synonymous variant, is considered benign. Let's check on the second variant. So in this case, we have K583R. Again, similar to before, we have a screenshot of what you would see if you were to search on this variant in Mastermind. And again, bringing your attention to the ACMG interpretation. This variant, based on the criteria, is classified as likely pathogenic. And then below that, again, looking at the ClinVar record, it's also considered pathogenic or likely pathogenic. In addition to that, below you see some curated clinical evidence. So again, looking at different types of information that's been curated within Mastermind. So in this section right here is the clinical evidence, and so this is just zooming in on that. And then if you needed additional information, you could certainly go to the reference to find that. All right, so now that we've had a chance to identify the pathogenic variant for NF1, we can then turn to the Cancer Knowledgebase, or CKB. So within CKB, we curate all different types of evidence related to variants, including actionable evidence, of course, and then prognostic, diagnostic, and risk factor. And so in this case, this is a germline variant, and so we wanted to see if there's any risk factor evidence related to NF1. And we can do this by searching at the gene level. So this is a screenshot of the web-based version of CKB Boost. And so on the left-hand side here, we've searched on NF1. And so you see some information related to the gene at the top, but below that is where you're going to see information related to, in this case, specifically we're looking for evidence related to risk factor. So here, we again, just zooming in, NF1, and if we actually filter down to risk factor through this column right here called evidence type, we can actually determine to see if there's anything related to, in this case, breast cancer, as well as neurofibromatosis type 1. And so what we see is that germline mutations in NF1 result in neurofibromatosis type 1 and are associated with increased risk of developing neurofibroma. In addition to that, you can see below, germline NF1 mutations are associated with increased risk of developing breast cancer. And so, and you can see the reference here is nccn.org. So these are recommendations through at the NCCN guidelines. So now that we've seen that there is an increased risk of developing breast cancer, which this patient does have, we can then move on to looking at the variants that were identified in BRCA1 and BRCA2 for this patient. And so you have the ability to search specifically by gene variant within CKB. So looking at this first BRCA2 variant, E2856A, once you find it in CKB, you can see the gene variant description, the protein effect. So this variant is functionally characterized within the literature. This evidence has been curated into CKB within the variant description, and there's all the references used to support that evidence. And so it's been identified, again, as no effect, behaves similar to wild type. If we look at the BRCA1 variant and search on this one, we see that this particular variant actually results in a loss of function. Again, if we focus on the variant description here, there is evidence to support that this variant results in a loss of protein function. So it's really that loss of function variant that we would be interested in order to understand, is there any actionable evidence? And of course, looking at below in these 5 different tabs, we can take advantage of what's called the extended evidence. So up here, we have what's called category variant paths. So this specific variant is a loss of function variant. And if we look at this first tab, variant evidence, there's no evidence specific to this variant within the literature. However, we can sort of walk up this path and leverage the category variant that this variant belongs to, which would be BRCA1 inact mute. And that's where you're going to find that evidence. That's within the extended evidence tab. So again, we can filter down to breast cancer in terms of the tumor type, and then figure out, okay, we filtered down, there are therapeutic options, just for example, this talazoparib, which is FDA approved. And then we can also see the efficacy evidence annotation that's associated with this. So just zooming in here, every actionable efficacy evidence annotation is basically a result, results summarized into one sentence. And so that will include information related to the primary endpoint or the secondary endpoints, and that's through the trial that was used to support that FDA drug approval. So in this case, this may be a treatment option for the patient, highlighting that this drug is FDA approved for patients with HER2-negative breast cancer, harboring, in this case, germline BRCA1/2 mutations, so perhaps it could be used off-label. All right, so let's just sort of wrap up this first case. So this patient, again, NF1 and BRCA1/2 variants were identified in this patient, and we used both Mastermind and CKB. One, in Mastermind, we found diagnostic insights for germline cancer risk variants, finding both a benign and pathogenic variant. You also see the ClinVar record as well. And then within CKB, we are able to find actionable insights for somatic variants. So based on tumor type info, treatment options, and then the trial results from that trial for that specific drug. All right. Moving on to case 2. So for case 2, we have a patient that has been diagnosed with castration-resistant prostate cancer, and this patient also has a family history of cancer. Specimens, similar to the previous case, tumor sample, and then whole blood sample for matched control. Samples were sequenced using whole exome sequencing for this one. So for results, this patient was found to harbor an MLH1 variant. This was identified as germline. And then somatically, there were 4 other variants with potential biological relevance. So let's focus on that MLH1 variant. So again, in this case, we're going to use Mastermind to interpret the germline variant with curated evidence. And we can see on the left-hand side here, the screenshot, when we search for this variant, R755W. And based on the ACMG interpretation that's been curated within Mastermind, we can see this variant is likely pathogenic. Previously, you saw that there was information related to ClinVar if the variant was found in ClinVar. In this case, this variant is not actually in ClinVar. Below that, you can also see some curated functional evidence. So again, this is taken from the right-hand side, functional evidence that was used to support this classification of likely pathogenic. All right, so now we're going to move to CKB. So we can actually look at cancer associations by tumor type and category variants. So I mentioned that the category variants before. This particular variant, we see that it's pathogenic, likely resulting in a loss of function. So if we actually go in through the gene detail page for MLH1, so we would search on MLH1, and then again looking at evidence, so that evidence tab right below the gene description at the top here, and then filter down to prostate cancer. So we can filter down to prostate cancer, and you can see that there is a combination therapy that is FDA approved for patients that harbor MLH1 inactivating mutations. In addition to that, there is a tab next to the evidence called clinical trials. So we curate clinical trials from clinicaltrials.gov, and on this page, you would see trials that include recruitment criteria specific to MLH1. And so you can see the active trial info per category variant here. All right. So for this patient that was diagnosed with castration-resistant prostate cancer, family history of cancer with pathogenic germline MLH1 variant. In addition to that, MLH1 is also linked to Lynch syndrome, which you may have seen within Mastermind on the ACMG criteria. So if we go through Mastermind and CKB, again, diagnostic insights for germline cancer variants, but also a variant that was not found in ClinVar. For CKB, actionable insights, we can search on category variant information specific to a tumor type, identify treatment options, and trial options as well. All right, case 3. So this case is a pediatric patient diagnosed with B-cell acute lymphoblastic leukemia. Tumor sample was used for sequencing, specifically whole genome sequencing. And what was identified was a fusion, so DUX4 IGH fusion. What can we find on this fusion? So first, going into CKB, we can search for specific actionable insights from extended evidence, specifically just searching on the fusion. So CKB does include fusions, and you can see here that this particular fusion results in a gain of function. There's information related to the evidence that's used to support that gain of function, and then also that it's been identified in B-cell acute lymphoblastic leukemia. And then below that, you can see those category variant paths. So this fusion belongs to DUX4 fusion as well as DUX4 rearrange. So we can then look at extended evidence specific to this fusion. So in this case, we're looking at the parent categories that this fusion belongs to, so DUX4 fusion, DUX4 rearrange, and we can see right here there is information related to prognostic guidelines. And so if we look over here to the right, we can see DUX4, childhood B-cell acute lymphoblastic leukemia. And then if we look down below, you can see that specific prognostic information. Patients with this fusion actually have a favorable prognosis, so perhaps in this case, treatment could be changed in terms of that prognostic information. Now, perhaps we would like to find more information regarding prognosis and its association with this fusion. So we could then take advantage of Mastermind and look at the indexed evidence for somatic variants, including specific actionable insights. So here we are on the looking up this particular fusion, DUX4, IGH. We can actually filter down within Mastermind, so we can use these advanced filter search. In this case, we want to specifically highlight anything that's considered prognosis or related to prognosis. And then once filtering down to this, we can see the indexed evidence for prognosis. And again, you have this wealth of information that is right at your fingertips, and you're able to just quickly go to the paper if you needed more information. All right. So this is, again, our last case, but just to highlight this wrap-up, this patient was found to have a DUX4 IGH fusion within Mastermind, diagnostic insights for somatic cancer variants in this case. So it includes fusion variants as well as prognostic insights. And then in CKB, prognostic insights for somatic variants. And this is at the level of the guidelines and specific to that particular tumor type, the B-cell acute lymphoblastic leukemia. All right, so now that we've gone through some cases, you've seen how you can leverage both CKB and Mastermind. We do have CKB available in 3 solutions. So CKB Core, this is our free version. You have access to 50 genes and all of the related content to those 50 genes. There's also CKB Boost, which is web-based as well, requires a subscription. In this case, you have access to just over 2,000 genes and all the related content. That also includes just over 47,000 variants curated in CKB. And those screenshots that I showed you previously were from CKB Boost. For users that are interested in downloading the content, we have CKB Flex. So this is a scalable and flexible content integration option for, again, those 2,000 genes. So all of the content that would be available in CKB Boost is also available in this downloadable version. For Mastermind, there's also 3 different plans as well. There's the basic plan, so this is free for genomic research. So you have the ability to search over 30 million papers by gene and or variant, and then you can prioritize your results and see highlighted genes and variants in the context of what you're searching for. The most popular version of Mastermind is the professional version, so this is ideal for clinical decision support. So you get to prioritize your results by relevance, all the features of basic, and then plus more. Search into the supplemental data. A lot of times you're going to find variants buried in the supplemental data. Or also use it for powerful advanced search capabilities. I showed you how you can filter down to just prognostic information that you want. And of course, unlimited searches, and article views, and then also you can export the bibliographic search results. And then finally, similar to CKB Flex, there is data licensing as well for Mastermind. So this enables automation services. So again, how can you streamline your workflow? By having the ability to download this data or this curated content, and that's available in a flat data file or API. All right, so just to wrap up, especially focusing on those learning objectives that Mark mentioned in the beginning, we know that labs have been clearly adopting whole genome sequencing and large gene panels, and there's huge benefits to that. But there's also limitations as well. You have a significant amount of data. So in that case, it's really essential to make sure you have accurate and reliable resources to aid in the interpretation as well as treatment decisions. And that's where CKB and Mastermind can come into play. And really using the two, they very much complement one another in terms of germline information as well as somatic information in CKB and the actionability. So if we look to the future, or looking at emerging trends with whole genome sequencing, One thing we just wanted to mention was that we're starting to see more and more studies coming out based on the identification of pharmacogenetic variants. And this, again, could inform clinical impact of variants associated with drug-induced toxicity. So again, uncovering these valuable insights for patients. How can we make sure that they're getting the most appropriate care? Obviously, we want to avoid any adverse side effects, and so this is where pharmacogenetics comes in. So with that, I would just like to thank you all for attending our workshop, and we're happy to answer any questions. Yes.
Thanks for the presentation. I'm curious, with the acquisition of Boston Genetics, how are patients now benefiting more with that than before? And how are providers benefiting from all the technology that Genome has to put into the reports that Boston Gene is now popping up for patients? Like, what's different? What's new? And the second question is, will you clarify the benefits of CKD versus OncoKD for somatic cancer?
We'll tag team. So I'll take the first one. I'll repeat the question is, how has the acquisition of Boston Genetics, a team of curators, about 100, benefited patient care or enhanced the offering of Genomenon?
For patients with Boston Gene who are getting NGS testing at Boston Gene.
It's I don't know.
Is Boston Gene connected with Boston Genetics?
No, they're separate. They're separate. I can answer the question even though it was in error. I mean, so what I would have said if I had understood it differently is scale. The answer is scale. It also comes back to what I said before about the calls being provisional. So that funnel problem that I talked about with the proliferation of evidence and then the tapering down to the actionability, we're curating the genome. We're doing that gene by gene, many hundreds of genes each month. We're allowing that information to be integrated into those workflows to allow people to see that information faster, more sensitively, so more of that data is presented. And as Kara showed, it's at their fingertips, so they can see that information at point of care when they're ready to make the diagnosis. So sorry about the confusion, but I think you can see how there is great benefit to the whole molecular diagnostic apparatus, not just one specific hospital system. So Kara, if you want to address CKB and OncoKB.
Sure. Okay. So that's a great question. So, I mean, CKB and OncoKB are similar in that you can identify actionability of variants, of course. I will say with CKB, there is more curated content. I mentioned we have over 47,000 variants, just over 2,000 genes. But what also distinguishes CKB from OncoKB is we also include evidence related to what we call complex molecular profiles. So often, especially when it comes to case studies that are published, a patient has multiple variants that are contributing to the response of the therapy. And so when we curate evidence in CKB, we incorporate that into one molecular profile. So that molecular profile is tied to the tumor type, the therapy, and then, of course, that summary. In addition to that, we also curate clinical trials from clinicaltrials.gov. And so we cover several countries around the world, and we're curating weekly with those trials, so importing any drugs that already exist in CKB, so adding those trials, and then any new drugs that are coming out. We also include a significant amount of preclinical evidence as well. So if you're interested in, drug development or research, specifically looking at resistance variants, things like that, that also, there's a significant amount of content related as well. So, any other questions?
If not, we're happy to meet you at the booth, 1233, I believe.
Yep.
So thank you for your attention.
Thanks.